Preparation of apoptotic cell death. First, culture charcoal-2 cell in 96 well plate to increase the cell density to 10 power 4. Second, for apoptosis cell death, the cell is treated with 2% hydrogen peroxide and add 0.5% DMSO to the normal cell. Then, incubate the cell at 37 degrees Celsius with 5% carbon dioxide for 24 hours. For RNA extraction. First, add triazole reagent into a cell pellet. Second, add 200 microliters of chloroform to a sample and mix by tapping the tube. Third, centrifuge the tube at 10,000 rpm for 15 minutes. After that, collect the colorless upper layer of the solution and move to a new tube. Fourth, add 500 microliters of coisopropanol to precipitate RNA and mix by invert the tube. Fifth, centrifuge again at 10,000 rpm for 10 minutes. Sixth, after centrifuge, discard the supernatant out of the tube. The edge of the tube can be dried by gently tapping on the paper towel. Seventh, add 75% of coethanol into the tube and centrifuge at 10,000 rpm for 5 minutes. 8. Discard the solution and dry the tube at room temperature. 9. After the tube is dry, add 30 microliters of deep sea water to dissolve the RNA. Preparation of 1% agarose gel electrophoresis. First, weigh 30 micrograms of agarose powder in a laminar flask. Second, measure 30 ml of TAE buffer in measuring glass. Third, add the buffer in agarose gel, then heat in microwave for 1 minute or until all agarose solutions are dissolved. Fourth, while waiting for the gel get warm, set the casting tray by put the comb on the tray. Fifth, after the gel gets warm, add 1.5 microliters of safe dye to the gel and mix by gently. Sixth, pour the gel solution to the tray and wait for 20 minutes. Seventh, after 20 minutes, the gel becomes hard. Take out the comb carefully from the tray and move the gel together with the tray to electrophoresis chamber. Make sure all of the gel in chamber is covered by TAE buffer. 8. For preparing sample, take 2 microliters of loading dye on paraffin paper. Then, followed by add 5 microliters of extract RNA. And mix the solution by pipetting on paraffin paper. and load into the agarose gel. Ninth, take 2 microliters of 1 kilobase pi DNA marker into agarose gel. Ten, close the chamber, turn on the machine, set, and run the machine for 20 minutes. The results after separate by using agarose gel electrophoresis are shown in the figure. Determination of regulate gene in apoptotic cell death by using RT-PCR. After RNA extraction, the RNA is reversed to single-strand DNA. The single-strand DNA is used as a template for synthesized double-strand DNA by using polymerase chain reaction or PCR. First, prepare the microtube placed on ice. Carefully, all of the reagents for this experiment should keep on ice. The reaction mixture of PCR consists of 4 reagents. First, 10 microliters of PCR master mix. Second, 3 microliters of forward and reverse primer. Third, 4 microliters of cDNA template. The total volume of this reaction is 20 microliters. The specific primer of this experiment are shown in the table. Second, add all of the reaction mixture to the microtube. Mix and spin down for 15 seconds. Third, turn on the PCR machine. Set the PCR condition as in the picture. Fourth, place the PCR tube in the machine and close the lid. Run the machine until it completes. After that, measure the PCR product by using 1.2% agarose gel electrophoresis. The results show as follows.